So we just looked at the idea of an amino acid molecule consisting of an amino group and a carboxyl group. And the carboxyl group, of course, can give away an H+, leaving a negative there. It gives away an H+, so it's an acid. That H+ can later join onto the amino group, and because the amino group is taking H+ ions out of solution, that means this part is a base. And every amino acid has that. But what every amino acid has that might not be the same is the R group. So the R group, there is no R in the periodic table. So R is used as something variable, something that could be anything. And this is what gives every amino acid its different properties. It's what means every amino acid will fit into a different enzyme and allow it to do something else. And we'll look at some examples here. I do have to emphasize that you do not have to know the structures of all the 20 amino acids that we're going to look at. And there are 20 amino acids because that's how many make up our proteins. There are only 20 different amino acids making up what is you. You are a protein structure with a bunch of other molecules around it. So let's have a look at the first one. And we've got here, well, we've got some non-polar groups. Non-polar groups, remember, are those that don't have a slight charge. They're not going to have a delta positive or a delta negative charge in them. So here we are. Here's the amino group. Here is the COOH, the carboxyl group. And here, the R group is simply a hydrogen. So this is the simplest of the amino acids. There it is. And this one's quite important because it's only little. It's only got one R group. That means that doesn't get in the way. And what I mean by not getting in the way, we'll see in a couple of videos' time. But yeah, that's your simplest amino acid. All you needed to do was add a hydrogen atom into the R group. Here's the next one. It's called alanine. So here we've got the amino group. Here we've got the COOH carboxyl group. Here we've got an H sticking out. And now the R group is a CH3. So there's a C in the middle and 3 H atoms sticking out of the bottom. Oh dear, and I've put that in there. And now, that's beautiful, I've made an alanine molecule. So now, I've made 2 amino acids, a glycine and an alanine. Okay? They are the simplest of the amino acids, and they're non-polar. So this part sticking out here, it's going to repel water, because there is nothing that can attract the water. So there's a hydrophobic bit. So a non-polar R group is hydrophobic. That's pretty important when the whole protein gets into water. So the next one we're going to look at is, I'll just get my basic amino acid, and it's cysteine. Now I'm interested in cysteine. It is a non-polar one, but the idea of it being a non-polar amino acid is superseded by the fact that it's got a sulfur atom in it. Now I don't have any sulfur atoms in my kit, so I've made the amino group here, the R group, out of this sulfur atom, which is like a balloon hastily stretched over an oxygen. So I used oxygen because it's got 2 holes, because it produces 2 covalent bonds. So here's my cysteine, and it's got that S there. There is another one that has that there, but this cysteine is vitally important. And there's a reason for that. When proteins form, they often get their shape by forming bonds between the R groups. So here's an R group. Sorry about this tail sticking out there. It's left over from when I stuck the balloon over that atom. So here's what happens. Here's 2 cysteines. And what happens is, whoa. Now, it's fallen apart, but you see what's happened there? The 2 sulfur atoms have formed a covalent bond. So, sulfur, just like oxygen, forms 2 bonds. And when we take those hydrogens off, then we get a covalent bond formed between them. That covalent bond is called a sulfur bridge, or sometimes a disulfide bridge, or you might see it as an S bridge. But whatever it is, it forms a covalent bond that can join 2 amino acids together. All right? The last thing I'll look at right now on this video is a larger group. So there's a larger non-polar group here. And you can see this polar group is only made of, this non-polar group, sorry, this R group, is only made of carbon and hydrogen atoms. That's a fairly non-polar bond. Because they have kind of equal electronegativities, we don't have a delta positive and a delta negative or any other kind of charge. So this will be a hydrophobic R group. And now, if I put that here, and you can see what it looks like here, we've got, oops, sorry, this one's upside down. Here's my amino group, NH2, or in this case, NH3+, because it's in water. Here's my carboxyl group, COO, and the H has been given off. And now up here there's a CH2, there's a carbon in the middle here, and two CH3s sticking out of there. It's a large R group, but it hasn't got any polar bonds in it. So this will be a hydrophobic R group. That means it's going to try and get away from water. That means when a protein is made with this in it, this is going to be on the inside of the protein, trying to be sheltered from the horrible water, because it's hydrophobic, by other R groups sticking around it. We'll do the others in a minute.